Around 225 million years ago, the first true mammals appeared on Earth. They were small, fur-covered, warm-blooded, and for the next 164 million years, their descendants lived alongside dinosaurs, not after them, alongside them. While giant predators dominated the daylight hours, our ancestors survived in the shadows, living fast, staying hidden, and rarely growing larger than a modern small mammal. Most people think the dinosaur extinction is what matters to our story, but an equally important story happened before that. Because surviving the age of dinosaurs came with a cost, somewhere during those millions of years, the mammalian lineage lost a DNA repair tool that almost every other vertebrate on Earth still carries today. Fish have it, birds have it, reptiles have it, amphibians have it. You do not. And scientists now believe that loss may be connected to how mammals age, how our cells accumulate damage, and why cancer is so common across our entire branch of life. Even stranger, when researchers restored the missing gene in laboratory mice, the machinery needed to use it was still there. After more than 100 million years, it worked almost immediately. The tool was gone. The blueprint for using it was not. This is not just a story about dinosaurs. It's a story about something your ancestors lost while living in their shadow, and why traces of that loss may still be active in your body right now. A long stretch of deep time existed before mammals became creatures of burrows, leaf litter, and night. To understand the inheritance carried by every mammal today, it helps to begin in a world where our lineage stood in a very different position. The story starts roughly 225 million years ago, during the late Triassic, when the first true mammals emerged from a broader group known as the cynodonts, themselves descendants of the synapsid lineage. The word synapsid rarely appears outside paleontology, yet these animals form the branch of life that eventually led to humans. Their history reaches far deeper than most people realize. Millions of years before the first dinosaur placed a foot on Earth, synapsids already occupied landscapes across the planet. Some were small, some large, and some held positions near the top of their ecosystems. One of the most familiar examples is Dimetrodon. Museum visitors often see its sail-backed silhouette displayed near dinosaurs and assume it belonged to the same group. In reality, Dimetrodon shared a far closer evolutionary relationship with you than with any dinosaur. It lived roughly 40 million years before the earliest dinosaurs appeared, representing an older chapter in the story of terrestrial vertebrates. The distance separating Dimetrodon from Tyrannosaurus was greater than the distance separating Dimetrodon from modern mammals. For a time, the mammalian branch seemed poised for success. Then another lineage began its rise. During the Triassic, the archosaurs expanded across the world. From this group came crocodilians, pterosaurs, and eventually the dinosaurs. Their diversification transformed ecosystems on a planetary scale. Forests, floodplains, deserts, and coastal environments gradually filled with forms that grew larger, faster, and increasingly dominant. The change was not sudden. No single catastrophe forced mammals into retreat. Across tens of millions of years, ecological opportunities narrowed. Larger body sizes became harder to sustain. Predation pressure intensified. Survival favored animals that could remain unnoticed, reproduce efficiently, and exploit resources overlooked by larger competitors. By around 200 million years ago, the mammalian lineage occupied a very different role than its ancestors had known. Most species were small, often comparable in size to modern shrews or rats. Many sheltered underground or within dense vegetation. Insects, seeds, and other modest food sources became staples of daily life. Lifespans were brief, and reproduction occurred early. Fossils preserve glimpses of these animals. Morganucodon, among the earliest true mammals, possessed a body plan that would have appeared familiar to anyone observing a modern insect eating mammal. Hadrocodium was even smaller, weighing only about 2 grams. Its delicate skull hints at a creature that could fit comfortably in the palm of a human hand. Then there was Repenimemus. Its discovery surprised researchers because it broke the expected pattern. About the size of a badger, it stood among the largest mammals of the entire Mesozoic era. Fossilized remains revealed fragments of a young dinosaur within its stomach contents, evidence that at least one mammal occasionally turned the tables on its enormous neighbors. Even so, Repenimemus illustrates the limits imposed on the lineage. A badger-sized hunter represented the upper edge of mammalian possibility for much of the dinosaur age. These creatures endured, adapted, and persisted through conditions that lasted for more than 160 million years. Their success kept the lineage alive, though the cost of that survival was accumulating in places no fossil could immediately reveal. A span of 164 million years is difficult to hold in the mind. Continents shifted their outlines during that interval. Mountain ranges rose and wore away. Entire ecosystems appeared, flourished, and vanished. Against that immense backdrop, the mammalian lineage spent almost the whole period following a single rule: wait for darkness. From roughly 230 million years ago until 66 million years ago, mammals lived primarily as nocturnal animals. Their active hours began when daylight faded, and many of the dominant hunters withdrew from movement. This pattern endured for a duration so long that modern humanity occupies only a tiny fraction of it. Our own species has existed for about 300,000 years. The nocturnal chapter lasted more than 500 times longer. The evidence does not come mainly from bones. Fossils reveal shapes, sizes, and habitats, yet they rarely preserve behavior. The stronger clues remain alive today, carried within the bodies of mammals across the world. Features of the eye, the organization of sensory pathways, and the structure of the brain all point toward ancestors adapted to life after sunset. Researchers have approached this question by studying living species rather than relying solely on extinct ones. In 2017, scientists examined behavioral data from 2,415 mammal species, representing every major branch of the mammalian family tree. Using those patterns, they reconstructed the probable lifestyle of the common ancestor from which modern mammals descend. The conclusion emerged with unusual clarity. That ancestor was nocturnal. The finding reaches beyond creatures that currently avoid daylight. Even mammals active beneath the midday sun retain traces of that inheritance. Their visual systems reveal an emphasis on gathering limited light. Their hearing and smell often carry levels of refinement uncommon among many other vertebrates. These are features shaped across immense stretches of evolutionary time, preserved long after the circumstances that produced them began to change. Daytime activity returned only gradually. Among the earliest groups to establish a substantial presence in daylight were the simian primates, the branch that includes monkeys and apes. This transition unfolded approximately 52 million to 33 million years ago. By then, the world that had constrained mammalian behavior for so long had already transformed. The dominant dinosaur predators were gone, ecosystems had reorganized, and opportunities unavailable for countless generations had finally emerged. Such timing reveals the depth of the bottleneck. Mammals did not rush into daylight the moment conditions shifted. Evolution works through inheritance, and inheritance carries momentum. Bodies shaped for darkness remain shaped for darkness until many generations gradually alter them. Throughout most of mammalian history, Survival depended on avoiding the hours when sharp eyed predators ruled the landscape. A small animal moving across open ground beneath daylight risked becoming part of another creature's meal before reproduction could occur. An animal that waited until darkness gained another chance to pass its genes onward. Repeated endlessly across 164 million years, that pressure became more than behavior, it became biology. It entered the architecture of senses, the wiring of brains, and the rhythms governing daily life. Long after the world changed, the imprint remained, woven into every mammalian lineage that followed. The loss can still be seen every time a mammal opens its eyes. Across forests, grasslands, mountains, deserts, and oceans, mammals move through a visual world that is less colorful than the one inherited by their distant vertebrate ancestors. The difference is so old that it feels natural, almost inevitable. Yet the story began during those long ages when survival depended on operating under conditions where color carried little value. The earliest vertebrates possessed a remarkably rich visual toolkit. Their eyes contained four distinct types of cone cells, each sensitive to different portions of the light spectrum. Together, these cells created a broader perception of color than most mammals experience today. Ultraviolet light, entirely invisible to human eyes, formed part of that world. Many fish still perceive it. Numerous reptiles retain it. Birds continue to navigate a visual landscape filled with hues and signals beyond human awareness. This ancestral arrangement persisted across vast branches of life. Fish kept it, amphibians kept it, reptiles kept it, birds kept it, mammals did not. As mammalian ancestors spent generation after generation active under low light, evolutionary priorities shifted. Color discrimination offers little advantage when illumination is scarce. Detecting movement becomes more important. Recognizing contrast becomes more important. Locating food, avoiding predators, and navigating terrain depend increasingly on sensitivity to faint light rather than the ability to distinguish subtle shades. Within the eye, another type of cell was already suited to that task. Rod cells excel at gathering limited light and detecting motion. Under nocturnal conditions, they became invaluable. Selection favored visual systems that performed well after sunset, and the balance between rods and cones gradually changed. While this process unfolded, some cone pigments ceased to matter. Genes survive through usefulness. When a gene no longer contributes meaningfully to survival or reproduction, mutations can accumulate without consequence. Across millions of years, two of the four ancestral cone pigments acquired enough changes to stop functioning altogether. Their loss was not sudden. It unfolded across spans of time so long that no individual animal would ever have noticed the transition. The consequences remain visible today. Most mammals are dichromats, meaning they rely on two primary cone pigments instead of four. Dogs experience the world this way. Cats do as well. Horses, mice, elephants, and countless others share the same limitation. Their perception is shaped largely around blues and yellows, while distinctions that humans recognize between reds and greens are greatly reduced or absent. Human vision often appears to contradict this pattern. Compared with many mammals, primates possess richer color perception. The path that produced it reveals how complete the earlier loss had been. Around 30 million years ago, a duplication of an existing pigment gene occurred within portions of the primate lineage. One copy continued performing its original role. The other gradually changed, creating sensitivity to an additional range of colors. What emerged was trichromatic vision, a useful innovation for identifying fruit, leaves, and social signals in daylight environments, yet it did not restore the original ancestral condition. The fourth cone pigment never returned. Ultraviolet perception remained absent. Evolution found a workaround, though the missing pieces stayed missing. Color offers one of the clearest records of the nocturnal bottleneck. The eyes of mammals still carry evidence that darkness shaped them for far longer than daylight ever has. Inside the cell, damage is often a matter of shape. A molecule bends where it should remain straight. Two neighboring pieces join where they should stay separate. The pattern is tiny, far smaller than dust, yet the consequences can follow a body for years. One of the oldest repair tools for this kind of damage is an enzyme called photolyase. It exists across an extraordinary range of life. Bacteria carry it, plants carry it, fungi carry it. Fish, amphibians, reptiles, and birds carry it. Across the living world, photolyase is so widespread that its absence in mammals becomes the strange fact, the gap where a common inheritance should be. Its work concerns damage caused by ultraviolet radiation. When ultraviolet light strikes deoxyribonucleic acid, it can fuse neighboring bases into structures called cyclobutane pyrimidine dimers. These fused places distort the molecule's shape. If the damage remains, the cell may copy the altered sequence incorrectly during division. Mutation enters the record. With enough such errors, tissue ages faster, and cancer becomes more likely. Photolyase performs its repair with a kind of molecular economy. It absorbs visible light, then uses that energy to split the abnormal bond and restore the original structure. Sunlight, the same force that helped create the injury, becomes the energy source used to undo it. The damaged place is reversed directly, without cutting away a section, without rebuilding a stretch of genetic material, without calling in a long chain of backup machinery. That directness matters. In many organisms, photolyase can repair this ultraviolet injury with an efficiency near 90%. The enzyme finds the distortion, binds to it, gathers light, and returns the molecule closer to its former state. For animals exposed to daylight across a lifetime, such a tool is more than convenient. It is a standing defense against one of the most constant sources of cellular harm on Earth's surface. Mammals lost it. During the Mesozoic era, while the lineage was shaped by night activity, the photolyase gene stopped being maintained in the same way. An enzyme that depends on light has little value in generations built around avoiding daylight. Mutations could gather inside the gene without strongly reducing survival. No great crisis marked the loss. It was the arithmetic of disuse, repeated through ancestry until a working tool became a broken remnant. The timing has been traced through phylogenomic analysis, the comparison of gene histories across many species. Photolyase relatives remain visible across the tree of life, yet mammals no longer carry the active ultraviolet repair version used so broadly elsewhere. The pattern places the loss within the mammalian story during the Long Age when dinosaurs dominated the daylight world. Mammalian cells were left with nucleotide excision repair. This system is real, important, and still life-preserving. It recognizes distorted deoxyribonucleic acid, cuts out the damaged stretch, rebuilds the missing sequence, and seals the repair. Its work is careful, though it requires more steps and more molecular coordination than photolyase. A direct reversal became a reconstruction job. Most damage can still be handled, yet some errors remain. Across years, across skin exposed to sunlight, across countless divisions of cells, those missed repairs become part of mammalian aging's deeper machinery. Consider a patch of skin exposed to sunlight during an ordinary afternoon. Nothing dramatic appears to happen. The surface warms. Cells continue their work. Yet deep within those cells, countless interactions unfold as ultraviolet radiation passes through living tissue. Some of that energy leaves no lasting trace. Some of it alters the structure of deoxyribonucleic acid itself. For most vertebrates, one line of defense waits specifically for that moment. Fish possess it, birds possess it, reptiles possess it. When ultraviolet damage forms certain molecular distortions, photolyase can identify the problem and reverse it directly. The injury is corrected before it has much opportunity to linger. Mammals must rely on a different strategy. When ultraviolet radiation creates damage inside mammalian cells, the burden falls upon the replacement system inherited after photolyase disappeared. Nucleotide excision repair patrols the genome, searching for abnormalities. Once a damaged region is recognized, molecular machinery removes a section of genetic material surrounding the defect. New material is then synthesized and inserted into the gap. The process works remarkably well, considering its complexity. Human life would be impossible without it. Complexity, however, creates opportunities for imperfection. Every additional step introduces time. Every additional step introduces the possibility that damage remains undetected, persists longer than it should, or is repaired with small inaccuracies. Most of the time, the system succeeds. A small fraction of failures becomes important because the number of repair events occurring across a lifetime is enormous. Each unrepaired lesion carries the potential to become a mutation. A mutation may pass harmlessly through a cell's history. Another may alter a gene involved in regulating growth. Another may affect mechanisms that normally prevent uncontrolled division. The progression is gradual, unfolding across years and decades, yet the chain of events is well established. Accumulated genetic errors can transform ordinary cells into cancerous ones. This helps explain why mammals display a particular vulnerability to ultraviolet-related skin cancers. Human medicine recognizes several forms, including melanoma, basal cell carcinoma, and squamous cell carcinoma. Their immediate causes differ, yet all arise within a biological context where ultraviolet damage is processed through a repair system less direct than the one retained by most other vertebrate lineages. The significance extends beyond cancer alone. Every cell carries a history of maintenance. Proteins must be repaired or replaced. Genetic information must remain stable. Damage must be recognized and corrected. Aging emerges partly from the gradual accumulation of imperfections within these maintenance processes. The loss of photolyase represents a specific example of a broader pattern, one in which long-term biological upkeep became less strongly protected across mammalian evolution. Researchers studying comparative aging have increasingly focused on this connection. Among them is Professor João Pedro de Magalhães of the University of Birmingham. In 2023, he proposed the Longevity Bottleneck Hypothesis, bringing together observations from genetics, cancer biology, and evolutionary history. Within that framework, photolyase occupies a special place because its disappearance can be traced directly. The missing repair tool is visible in the genomic record. What makes the story striking is its clarity. Many evolutionary events leave ambiguous traces. This one left a molecular absence. A repair system once shared broadly across life vanished from the mammalian lineage, and the consequences continue to echo through every generation that followed. The repair shop remained open, yet one of its most efficient tools was removed from the bench. Imagine two animals born into the same dangerous world. One directs more of its energy toward rapid growth, early reproduction and producing offspring as soon as possible. The other invests more heavily in cellular maintenance, long-term repair, and a body designed to remain healthy deep into adulthood. In a landscape where few individuals survive very long, only one strategy consistently wins. This outcome sits at the center of evolutionary biology. Natural selection rewards traits that increase the likelihood of passing genes into the next generation. Beyond that point, its influence weakens dramatically. An adaptation that helps an animal survive until reproduction can spread rapidly through a population. A trait whose benefits appear decades later may receive little attention from selection if most individuals never live long enough to experience those benefits. For the mammalian lineage, during the age of dinosaurs, this principle operated continuously. Predators, disease, injury, hunger, and environmental hazards removed countless individuals long before old age became relevant. Life unfolded on compressed timescales. Reproduction arrived early. Mortality arrived early as well. Under such conditions, energy became an evolutionary budget. Every biological function carried a cost. Repairing damaged tissues required resources. Maintaining highly efficient cellular defense systems required resources. Supporting mechanisms that protected a body far into the future required resources. Those resources could alternatively be directed toward producing offspring sooner or in greater numbers. Generation after generation, selection favored the traits most useful in the immediate struggle to survive and reproduce. A mammal that matured quickly and left descendants contributed more to future populations than one that invested heavily in longevity, yet failed to reproduce before death. The arithmetic repeated across millions of years. This helps explain why the disappearance of protective systems could occur without immediate evolutionary consequences. A repair pathway whose greatest value appears late in life provides little advantage if few individuals reach that stage. A regenerative capability that requires extended recovery time contributes less when predators routinely shorten lives before recovery becomes relevant. Functions associated with long-term maintenance gradually lose their protective importance when the future itself becomes uncertain. Professor João Pedro de Magalhães describes this process through the concept of the longevity bottleneck. The phrase does not refer to a single disaster or sudden genetic collapse. It describes a prolonged period during which selection repeatedly favored fast life histories while placing weaker pressure on mechanisms that support exceptional longevity. The outcome was cumulative. Each generation inherited the biological consequences of the one before it. Small shifts accumulated. Repair systems became less central. Reproductive efficiency became more central. The balance between maintenance and reproduction tilted in one direction for an extraordinary span of time. An important detail often goes unnoticed. Evolution does not plan ahead. It cannot preserve a trait because that trait might become useful millions of years later. Selection acts within the conditions of the present. During the Mesozoic world, immediate survival carried greater weight than distant possibilities. When the dinosaurs disappeared, mammals entered environments filled with new opportunities. Body sizes expanded, ecological roles diversified, new forms emerged across the planet. Yet evolution could only work with the inheritance already present. The biological architecture shaped during those long ages remained embedded within the genome. The losses endured because the forces that produced them operated for so long. By the time conditions changed, the pattern had become part of what a mammal was. A heartbeat is an act of maintenance. Each contraction moves nutrients, oxygen, hormones, and heat through a living body. In mammals, that work never pauses. Day and night, youth and old age, the machinery remains active, sustaining an internal temperature that stays largely independent of the surrounding world. This ability, known as warm-bloodedness, was among the most important advantages ever acquired by the mammalian lineage. During the long ages, when darkness offered safety, a stable internal temperature opened opportunities unavailable to many competitors. While reptiles depended more heavily on environmental warmth to reach peak activity, mammals could remain energetic after sunset. They could search for food, travel, and reproduce during hours when much of the landscape operated under different constraints. The advantage was substantial, yet it came attached to a permanent energetic bill. Heat does not appear freely. Every degree of internal warmth requires fuel. Food must be gathered, digested, processed, and converted into energy. The body becomes a furnace that continually consumes resources simply to remain itself. For small mammals, the demands become especially intense. A shrew lives near the edge of its metabolic limits. Food is transformed into energy almost as quickly as it is acquired. Missing meals carries serious consequences because the body has little room for interruption. The engine runs at extraordinary speed, supporting survival in a challenging world while simultaneously imposing relentless physiological pressure. Activity at this scale reaches far beyond movement alone. Cells must divide, proteins must be built and replaced, damaged structures must be repaired. Every cycle of replication introduces opportunities for mistakes. Biological systems contain safeguards, proofreading mechanisms, and repair pathways, yet no process achieves absolute perfection. Tiny errors accumulate across time, scattered through tissues and organs like faint marks on a manuscript copied repeatedly through generations. Researchers have long observed a broad relationship between metabolic intensity and lifespan among mammals. Species with extremely rapid mass, specific metabolic rates, often experience shorter lives. Mice and shrews mature quickly, reproduce quickly, and age quickly. At the opposite end stand animals such as the bowhead whale, whose metabolism operates at a more measured pace relative to body size. Some individuals survive beyond 2 centuries, carrying cellular systems that endure far longer than those of their smaller relatives. The pattern is not absolute, and biology always contains exceptions, yet the underlying connection remains difficult to ignore. A body that processes energy at extraordinary speed generates more opportunities for molecular wear. More cellular activity means more replication events. More replication events create additional chances for mutation. Across decades, these differences become increasingly significant. Viewed through the lens of mammalian history, warm-bloodedness begins to resemble a bargain shaped by deep time. Survival during the nocturnal era depended upon mobility, awareness, and independence from environmental temperature. Those benefits carried immense value. Without them, many mammalian ancestors might never have persisted through the age of dinosaurs. The cost accompanied the benefit from the beginning. Every mammalian cell inherited an energetic lifestyle that demanded continuous expenditure. The same internal furnace that opened the night also accelerated the passage of biological time within tissues. Survival gained a powerful ally, while aging acquired another source of momentum woven into the metabolism of the lineage itself. A faint scent drifting through leaf litter can carry more information than a distant shape glimpsed through darkness. For millions upon millions of years, that simple reality governed the lives of mammalian ancestors. Vision remained useful, yet survival increasingly depended on senses that could function when light offered little help. As the nocturnal bottleneck tightened its hold, mammals entered a world where smell became a primary source of knowledge. Chemical traces lingered on the ground, clung to vegetation, and hung in moving air. Food, rivals, mates, offspring, and predators all left signatures that could be detected and interpreted. The brain regions responsible for processing scent expanded accordingly. Neural tissue is expensive to maintain, and evolution rarely enlarges it without persistent pressure. In mammals, that pressure endured for 164 million years. Hearing underwent a similar transformation. Nighttime environments reward the detection of subtle sounds. A rustle, a footstep, a distant movement through brush, each could contain life-preserving information. Mammalian auditory systems became increasingly refined, capable of distinguishing details that would have escaped notice in many earlier vertebrate lineages. Sound could reveal what sight could not. A world that appeared uncertain through the eyes remained rich with information through the ears. Those sensory streams did not operate independently. The challenge facing a small nocturnal mammal was not merely detecting information, but combining it rapidly. Odors, sounds, touch, memory, and spatial awareness had to be integrated into coherent decisions. The nervous system therefore faced demands extending beyond raw sensation. It needed to compare signals, evaluate possibilities, and respond with speed. Evidence of this transformation appears in fossils. One of the oldest known mammalian brain endocasts belongs to Triconodon mordax, a Jurassic mammal that lived roughly 150 million years ago. Although the animal itself was modest in size, the internal shape of its skull reveals a forebrain that was already enlarged relative to many reptilian contemporaries of similar body mass. The trend had begun long before the age of mammals arrived. Within mammalian brains, the olfactory bulbs, cerebellum, and developing neocortex expanded along pathways consistent with the demands of nocturnal existence. The neocortex proved especially important. This layered outer region became a center for integrating sensory information, storing learned experiences, navigating environments, and adapting behavior. Generation after generation, evolutionary pressures favored individuals capable of processing complex streams of information with greater flexibility. An extraordinary paradox emerges from this history. The same conditions that stripped away biological capabilities also encouraged the growth of an organ unlike any previously seen on Earth. Color perception narrowed. Molecular repair systems disappeared. Mechanisms associated with longevity weakened. Alongside those losses, cognitive capacity continued to expand. The outcome would not become fully visible until much later. From that expanding neocortex eventually emerged symbolic thought, language, mathematics, music, philosophy, science, and the ability to reconstruct events separated from the present by hundreds of millions of years. A lineage compressed into darkness developed the neural architecture required to understand darkness itself. The brain following this story tonight carries that inheritance. Its structure reflects pressures imposed during an age when survival depended on interpreting scents, sounds, and fragments of information beyond the reach of sight. The same evolutionary forces that shaped mammalian limitations also helped construct the biological machinery capable of recognizing them. Some lineages carry the past with unusual completeness. They are living bodies, yet they also function like biological archives, preserving traits that vanished elsewhere while the mammalian branch was being reshaped by darkness, haste, and predation. To see the scale of what mammals lost, it helps to look toward animals that followed another path. The tuatara lives only in New Zealand, and it belongs to an order called Rhynchocephalia. Its relatives once spread much more widely, yet today the tuatara remains as the last surviving representative of that old branch. Its lineage separated from the ancestors of lizards and snakes around 250 million years ago, before the first dinosaur appeared. That history matters because the tuatara did not pass through the same mammalian compression. It retained a different relationship with time. In 2022, researchers examined tuatara aging through deoxyribonucleic acid methylation, a chemical pattern often used as a biological clock. The results showed exceptionally slow aging compared with mammals of similar size. Mortality does not appear to rise sharply with age in the mammalian pattern. Reproduction can continue across lifespans that would be extraordinary for small or medium-sized mammals. One male tuatara at the Southland Museum in New Zealand became especially famous after breeding successfully at 111 years old. Turtles offer another window into preserved maintenance. A study published in the journal Science in 2022 examined 52 turtle species and found that roughly 75% showed negligible or near negligible senescence. In those species, the likelihood of death does not climb steeply with age, and reproductive ability can remain stable across long stretches of life. Their bodies do not follow the same steep curve that defines most mammalian aging. Crocodilians also reveal the power of a slower biological rhythm. Many species can live 70 to 100 years, with reproduction continuing across much of adulthood. Their bodies repair injury, maintain muscle, and preserve function in ways that differ sharply from short-lived mammals. They are hardly immune to damage, disease, or death, yet their aging pattern follows another tempo. Among amphibians, salamanders show an even more visible form of retained capacity. Axolotls, native to Mexico, can regrow entire limbs with bone, muscle, nerves, blood vessels, and skin restored into working form. Some salamanders can regenerate parts of the heart, sections of the spinal cord, and tissues of the eye. These are not cosmetic repairs. They are organized reconstructions of living structure. Such animals are sometimes described as primitive, though that word distorts the lesson. They are not unfinished versions of mammals. They are successful lineages with biological tools that mammals no longer possess in the same form. In the specific realm of aging, repair, and long-term tissue maintenance, they reveal possibilities that were once more widely available across vertebrate life. Their bodies show what uninterrupted maintenance can look like when evolution does not repeatedly favor speed over durability. The comparison is biological, written in tissues that keep rebuilding while mammalian tissues decline. Beside them, mammals appear less like the inevitable peak of animal design and more like survivors carrying the marks of a long historical narrowing. Cancer leaves a different kind of fossil record. It is found inside bodies rather than stone, in cells that have copied themselves too many times with too many mistakes preserved along the way. When scientists compare cancer across vertebrates, mammals appear with a pattern that fits the deeper history already unfolding. The Longevity Bottleneck Hypothesis makes a clear prediction. If mammalian ancestors spent 164 million years under conditions that weakened long-term maintenance, then mammals should carry a higher burden of diseases linked to failed repair. Cancer belongs at the center of that prediction because it begins when cellular safeguards no longer hold the line. Large comparative studies have begun to show exactly that pattern. Researchers examining necropsy records across vertebrate groups found that mammals had the highest prevalence of both benign and malignant tumors. Reptiles followed behind them. Birds and amphibians showed lower rates. The pattern did not rest on one species or one habitat. It appeared across broad branches of the vertebrate tree. A 10-year study led by Arizona State University analyzed more than 16,000 necropsy records from 292 vertebrate species. Once again, mammals stood at the top. The numbers gave the pattern texture. Ferrets showed tumor rates reaching as high as 63%. Opossums and hedgehogs also carried high risks. These animals live with fast reproductive rhythms, short generation times, and bodies shaped by the same evolutionary pressure that favored immediacy over long repair. The connection is mechanical as much as historical. Cancer begins when cells accumulate mutations that alter the rules of growth. A tumor suppressor gene may lose function. A growth-related gene may become overactive. A cell may stop obeying signals that normally limit division. One change often does little. Several changes, gathered across time, can transform a normal cell into a lineage of cells that expands without proper restraint. That pathway becomes easier to enter when repair systems miss damage. The lost photolyase pathway is one example, especially in tissues exposed to sunlight. Ultraviolet injury that other vertebrates can often reverse directly must be handled in mammals by slower replacement machinery. Most damage is corrected. Some remains. Across years, those small leftovers become part of the mutation burden. Cancer, then, is not only a modern medical problem, it is also an evolutionary signal. A body built from imperfect maintenance systems must continually manage the consequences of accumulated error. Mammals developed many defenses against tumors, including immune surveillance, cell cycle controls, and programmed cell death. These defenses are powerful, yet they operate inside a biological framework already shaped by earlier losses. Professor João Pedro de Magalhães has suggested that the elevated cancer frequency in mammals may be one of the clearest living expressions of the longevity bottleneck. The idea does not claim that every tumor traces to one missing enzyme or one ancestral event. It points to a broader inheritance, a lineage whose maintenance architecture was narrowed across deep time and then carried forward into every descendant. The statistics, therefore, become more than clinical records. They are traces of old selection pressures, still visible in living tissue. Each tumor begins in the present, inside one body, yet the vulnerability behind it reaches back to a world where lasting repair mattered less than surviving long enough to reproduce. A layer of rock only a few centimeters thick marks one of the sharpest boundaries in the history of life. Beneath it lies a world ruled by familiar giants. Above it begins a different Earth, one reorganized by a single event that unfolded with astonishing speed. 66 million years ago, an asteroid approximately 10 kilometers in diameter, which is about 6.2 miles, struck the region now known as the Yucatan Peninsula. It arrived at roughly 20 kilometers per second, which is about 12.4 miles per second. The impact released energy beyond ordinary comparison. Rock vaporized. Material was blasted high into the atmosphere and beyond. Across enormous distances, the sky itself became part of the catastrophe. The first hours carried consequences on a planetary scale. Ejected debris arced through space before falling back toward Earth. As countless fragments re-entered the atmosphere, they heated the air and ignited widespread wildfires. Pressure waves raced outward from the impact zone and circled the globe. Tsunamis crossed entire ocean basins. Geological records preserve traces of these events on continents separated by thousands of kilometers, which is about 621 miles for every 1,000 kilometers. Those immediate disasters were only the beginning. Dust, sulfur rich particles, and fine debris accumulated in the upper atmosphere. Sunlight reaching the surface diminished dramatically. Food webs built upon photosynthesis began to fail. Forests, coastal systems, and marine ecosystems entered a period of cascading collapse. Conditions varied from place to place, yet the overall pattern was unmistakable. A biological crisis was unfolding across the entire planet. The Cretaceous Paleogene extinction eliminated approximately 75% of Earth's species. Every non avian dinosaur disappeared. Pterosaurs vanished from the skies. Mosasaurs disappeared from the seas. Countless plants, invertebrates, and marine organisms were lost as ecological networks unraveled. Communities that had persisted for millions of years were dismantled within what geologists regard as an instant. For mammals, however, the removal of dominant daytime predators changed the structure of possibility itself. The pressure that had shaped generation after generation was no longer present. Ecological space opened across forests, plains, river systems, and coastlines. Opportunities that had remained inaccessible for over 100 million years suddenly became available. The expansion that followed ranks among the most dramatic adaptive radiations known. During the 10 million years after the extinction, which is a brief interval in geological history, mammals diversified into an extraordinary variety of forms. Body sizes increased rapidly, feeding strategies multiplied, new lifestyles emerged. Some lineages returned to the oceans and eventually produced whales. Others took to the air and became bats. Large herbivores appeared. Predators evolved to fill abandoned roles. Primates joined the growing tapestry. Fossil records reveal that maximum mammalian body size rose along a steep trajectory after the extinction, eventually approaching new ecological limits around 40 million years ago. Creatures descended from ancestors, once confined to modest dimensions, were now producing animals comparable in size to modern buses. Yet every innovation unfolded within bodies already shaped by deep history. The lost visual pigments had vanished long before. Photolyase had disappeared long before. Biological systems connected to regeneration and long-term maintenance had already been altered. The descendants entering Paleocene sunlight inherited both opportunity and constraint. The gates had opened, but the travelers passing through them carried the marks of the road behind them. The disappearance of a pressure does not erase the shape it leaves behind. Mountains remain after tectonic forces fade. River valleys persist after the waters that carved them have changed course. Biology follows a similar rule. Once evolution has rewritten a lineage across immense spans of time, those changes do not vanish when circumstances improve. After the Cretaceous Paleogene extinction, mammals entered a transformed world. Dominant daytime predators were gone. Ecological opportunities stretched across continents. Forests, floodplains, coastlines, and recovering ecosystems offered space that had been unavailable for countless generations. Yet the earliest chapters of this new age reveal a surprising pattern. Most mammals did not rush into daylight. The Paleocene, spanning roughly 10 million years after the extinction event, was populated largely by small, adaptable mammals. They explored new diets, new habitats, and new ways of living. Even so, many remained active during darkness, twilight, or the edges of dawn and dusk. The external barrier had fallen away, but internal adaptations remained. Eyes, brains, sensory priorities, and behavioral patterns still reflected a heritage formed under very different conditions. Evolution works through inheritance before innovation. Every generation begins with what came before. Traits can be modified, expanded, or reduced, yet each change must proceed from existing biological structures. Mammals entering the Paleocene did not start with the sensory toolkit of daytime reptiles or birds. They carried the equipment assembled during 164 million years of nocturnal survival. Evidence from living species and fossil reconstructions suggests that truly daytime adapted mammals remained uncommon for a considerable period. Significant numbers of strictly diurnal species appeared later, particularly during the Eocene, between approximately 55 million and 33 million years ago. By then, entire ecosystems had already been reshaped. Forest canopies had diversified, mammalian groups had spread across the globe. Only gradually did some lineages begin making a lasting commitment to daylight activity. Among the most important of these were simian primates. As certain primate ancestors shifted into brighter environments, natural selection favored improved color discrimination. A gene duplication event produced a third cone pigment derived from an existing one. This adaptation allowed trichromatic vision to emerge, enhancing the ability to distinguish fruits, leaves, and social signals under daylight conditions. Even this achievement carried the signature of history. The recovered visual richness was incomplete. Primates did not regain the full ancestral visual system once possessed by early vertebrates. The fourth cone pigment remained absent. Sensitivity to ultraviolet wavelengths remained absent. What emerged was an ingenious reconstruction built from available materials, not a return to an earlier state. The broader mammalian story follows the same pattern. Photolyase did not return. Earlier regenerative capacities did not return. Biological systems associated with long-term maintenance did not return. Evolution moved forward using the framework that survived, adapting and modifying it while carrying inherited limitations into each new age. That inheritance remains visible today. Rodents, the most diverse mammalian order, are predominantly nocturnal. Bats, among the most successful mammalian radiations, conduct their lives almost entirely after sunset. Countless other species continue to occupy schedules shaped by rhythms established deep in the Mesozoic world. Humans stand somewhat apart within this pattern. As diurnal primates, we embraced daylight, then extended our activity beyond it through fire, lamps, and electric illumination. Yet the architecture of the mammalian body still bears older instructions. The bottleneck never truly ended. Its imprint traveled forward, generation by generation, and remains present in every mammal alive today. Scientific ideas often begin as scattered observations. A reptile ages differently from a mammal. A salamander restores tissues that a mouse cannot. Birds and reptiles retain biological tools that disappeared from the mammalian lineage. For decades, these findings accumulated across separate fields, each illuminating a small corner of a larger pattern that remained difficult to see in full. The pattern became clearer through the work of Professor João Pedro de Magalhães, a molecular biogerontologist at the Institute of Inflammation and Aging at the University of Birmingham. His research has long focused on one of biology's most persistent questions: why different animals age so differently despite sharing many of the same cellular foundations. In November of 2023, a paper published in the journal Bioessays proposed a framework that gathered many of these observations into a single evolutionary narrative. Known as the Longevity Bottleneck Hypothesis, it suggests that mammalian aging cannot be understood solely through modern ecology, physiology, or medicine. The roots of the problem reach far deeper, into the prolonged era when mammals survived under the dominance of dinosaurs. The hypothesis builds upon an earlier and widely supported concept known as the nocturnal bottleneck hypothesis. That framework argues that the ancestors of modern mammals spent an extended portion of their evolutionary history living primarily in darkness. Evidence for this history remains visible throughout mammalian biology, from sensory anatomy to patterns of brain organization. Professor de Magalhães extended the argument into a different domain. If nocturnal life reshaped mammalian senses and behavior, it may also have reshaped the machinery responsible for maintenance, repair, regeneration, and longevity. Viewed through this lens, many otherwise disconnected observations begin to align. Mammals lost photolyase, while other vertebrates retained it. Mammals generally possess weaker regenerative capacities than many reptiles, amphibians, and fish. Cancer appears more frequently across mammalian species than in several other vertebrate groups. Aging itself often proceeds along a faster trajectory than would be expected from body size alone. Each observation stands independently, supported by its own evidence. The hypothesis proposes that they may also share a common evolutionary origin. Among the strongest pieces of evidence is the disappearance of photolyase. This was not a speculative event reconstructed from indirect clues. Comparative genomic studies have traced the loss within the mammalian lineage. The timing aligns with the prolonged interval during which mammalian ancestors lived under the ecological constraints imposed by dinosaur-dominated ecosystems. A molecular change, an evolutionary context, and modern biological consequences appear linked within the same historical sequence. Professor de Magalhães also highlights the absence of regenerative abilities that remain widespread elsewhere among vertebrates. Salamanders regenerate limbs. Numerous fish species restore damaged tissues with impressive effectiveness. Mammals retain fragments of these capacities, yet the broader systems appear diminished. Such losses fit the prediction that long-term maintenance mechanisms could erode when immediate survival and rapid reproduction dominate evolutionary priorities. The longevity bottleneck hypothesis remains a hypothesis. Scientists continue examining its predictions, testing its assumptions, and searching for additional evidence. That process is essential to how science advances. Even so, the idea offers something rare: a coherent explanation capable of connecting genetics, aging, cancer biology, regeneration, and deep evolutionary history within a single framework. Across the long arc of the mammalian story, it provides a name for a pattern that had been waiting in the data for a very long time. A clue arrived from a place far removed from prehistoric forests and vanished worlds. It emerged under laboratory lights, inside carefully monitored cages, where researchers were investigating a question that reached deep into mammalian history. If a lost biological tool were returned to a mammal, would the body still recognize how to use it? The tool in question was photolyase, the light-powered repair enzyme absent from every living mammal. For countless generations, mammalian cells had functioned without it. The gene had disappeared long ago, carried away during the long era when daylight offered little advantage and survival depended on different priorities. What remained unknown was whether the loss had extended beyond the gene itself. Perhaps the surrounding cellular machinery had also faded through evolutionary time, leaving no pathway back. To test that possibility, scientists introduced the photolyase gene into laboratory mice. The experiment was straightforward in design, but profound in implication. Researchers were not merely observing whether the enzyme could be manufactured. They were asking whether a mammalian cell, separated from photolyase by more than 100 million years of evolution, could still operate the system correctly. The answer arrived with surprising clarity. Once the gene was present, the enzyme functioned. It recognized ultraviolet damage. It performed repair. It interacted with cellular processes in the manner expected from organisms that had never lost it. No extensive rewiring was required. No cascade of missing components had to be rebuilt. The molecular environment necessary for photolyase activity remained available. That finding revealed something unexpected about evolutionary loss. The disappearance of photolyase had not involved the removal of an entire biological network. Much of the surrounding framework endured. The missing element was the gene itself, while many associated pathways remained embedded within mammalian cells, preserved across immense spans of time. The consequences became visible in the mice carrying the restored enzyme. Compared with unmodified animals, they accumulated fewer ultraviolet-induced genetic alterations. Their tissues showed stronger protection against damage produced by sunlight. Rates of skin tumor formation declined. Molecular injuries that would normally persist were repaired before they could contribute to longer chains of cellular error. Viewed through the lens of the longevity bottleneck, the experiment carries unusual significance. Earlier examples traced losses that seemed permanent, features erased so long ago that they became part of the mammalian condition. Photolyase offered a different picture. Here was a vanished capability that could still operate when returned, suggesting that some evolutionary doors may be less completely closed than they first appear. Researchers have already begun exploring practical applications. Photolyase containing skin treatments are being investigated as a way to supplement the repair capacity that mammals naturally lack. The goal is not to rewrite mammalian biology, but to borrow a mechanism retained across much of the living world and place it back into service where it once existed. The experiment leaves behind an intriguing possibility. Deep within mammalian cells, traces of older biological architectures may still remain, preserved like foundations beneath a later structure. Photolyas demonstrated that one lost tool could function again when restored. The lesson extends beyond a single enzyme. It suggests that evolution sometimes removes a key while leaving much of the lock intact, waiting through ages longer than any mammal could ever remember. Somewhere within the mammalian family tree, a handful of species seem to move to a different rhythm. Their hearts beat, their cells divide, their lives unfold, yet the familiar mammalian timetable appears stretched. Years accumulate where they should not. Decades pass where biology predicts far less. These animals do not stand outside the story of the longevity bottleneck. They illuminate it. At first glance, the bowhead whale appears to challenge the entire idea. Living in Arctic waters, this giant mammal can survive for more than 2 centuries. Individuals born before major industrial changes in human society may still be alive today. Such longevity seems difficult to reconcile with a lineage supposedly burdened by compromised maintenance systems. Closer examination reveals a different picture. Bowhead whales carry additional copies of genes associated with deoxyribonucleic acid damage response and cellular regulation. Their tissues possess unusually effective mechanisms for preventing uncontrolled cell growth. Evolution did not restore what vanished during the bottleneck. It added new layers of protection on top of an inherited limitation. A similar lesson emerges from one of the strangest mammals alive. The naked mole rat weighs little more than a thumb, spends nearly its entire life underground, and routinely survives for around 30 years. A mouse of comparable size often lives only 2 years. Such a difference cannot be explained by body mass alone. Studies have shown that naked mole rats possess highly efficient systems for identifying and removing damaged proteins before those proteins accumulate inside cells. Their tissues also contain a distinctive form of hyaluronan, a structural molecule associated with resistance to tumor formation. Longevity appears to arise from enhanced maintenance of cellular quality, not from recovery of lost ancestral repair pathways. Bats offer another variation on the same theme. Among mammals, small size is usually paired with short life. Brandt's bat breaks that expectation dramatically. Weighing roughly 7 grams, it can survive for more than 40 years. Researchers studying bat biology have found evidence of unusual control over inflammation, efficient repair responses, and metabolic patterns that reduce the pace of accumulated cellular injury. The biological engine still carries the marks of mammalian history, yet it runs under a different set of adjustments. Across these examples, a consistent pattern begins to emerge. Long-lived mammals achieve their success through compensation. Each lineage discovers a different route. One strengthens protection against genetic damage. Another improves protein maintenance. A third alters metabolism or cellular signaling. The destination appears similar, while the pathways vary widely. That pattern is important because it reveals what these species have not done. None recovered photolyase. None regained the regenerative abilities common among certain amphibians. None escaped mammalian ancestry. The underlying constraints remained in place while evolution searched for alternative solutions. Seen from this perspective, the longest-lived mammals become evidence for the bottleneck rather than exceptions to it. Their biology resembles a structure reinforced over many generations, strengthened wherever weakness appeared, modified wherever damage accumulated. Longevity emerged through adaptation, layered upon adaptation. The remarkable ages reached by whales, bats, and naked mole rats do not erase the inherited boundaries of mammalian life. They reveal how much evolutionary innovation is required to push against those boundaries, and how persistent the original constraints have remained across the ages. Aging science has spent decades naming the ways a mammalian body wears down. Each name describes a different failure, a different layer of biology losing precision with time. At first, these failures appeared like separate problems scattered across cells and tissues. Through the lens of the longevity bottleneck, they begin to look more connected. One hallmark is genomic instability, the gradual accumulation of damage in deoxyribonucleic acid. Every cell depends on the accuracy of its genetic record. When that record is injured and repair does not fully succeed, small errors remain. Photolyase loss made one pathway of repair unavailable to mammals, while nucleotide excision repair carried the burden through a slower route. The body still protects itself, but protection leaves gaps, and gaps accumulate. Another hallmark is telomere shortening. Telomeres are protective caps at the ends of chromosomes, and they become shorter as cells divide. In fast-living mammals, especially small mammals with high metabolic rates, cell turnover can be intense. More divisions mean more opportunities for those caps to erode. When telomeres become too short, cells may stop dividing or enter states that contribute to tissue decline. Epigenetic drift adds another layer. Chemical marks that help regulate which genes are active and which are restrained change with age. These marks do not rewrite the underlying genetic sequence, yet they influence how cells behave. A liver cell, a skin cell, and a nerve cell all depend on stable patterns of gene regulation. As those patterns loosen, tissues lose some of the order that allowed them to function cleanly. Protein quality control also declines. Cells constantly build, fold, repair, and remove proteins. When that system falters, damaged or misfolded proteins begin to gather. Mammals with exceptional longevity often strengthen this machinery, which shows how important it is. In ordinary mammalian aging, however, the burden grows with time, and tissues become less able to clear their own molecular debris. Mitochondrial dysfunction belongs to the same pattern. Mitochondria are the structures that help cells convert fuel into usable energy. A warm-blooded body depends heavily on them. As they lose efficiency, energy production becomes less stable and damaging byproducts increase. The same internal furnace that helped mammals survive the night leaves cells with a lifetime of energetic strain. Cellular senescence gives the story another turn. Damaged cells can stop dividing as a defense against cancer. That response may protect the body in the short term. Across a longer life, senescent cells remain metabolically active and release signals that promote inflammation. A useful emergency brake becomes a source of chronic wear when the body lives far beyond the timeline evolution once favored. Stem cell exhaustion and disrupted cellular communication complete the pattern. Tissues need fresh cells for renewal, and organs need clear signals to coordinate repair. As these systems weaken together, aging becomes systemic. The bottleneck does not explain every detail, yet it gives these hallmarks a shared historical frame. Mammalian bodies are running maintenance programs shaped for short lives, now stretched across decades they were never strongly selected to sustain. A human life now often stretches far beyond the boundary that shaped most of mammalian history. Years accumulate past childhood, past parenthood, past the age when earlier survival would have counted as extraordinary. The body continues forward, carrying systems that were never strongly selected to endure for so long. For the overwhelming majority of mammalian evolution, life beyond reproduction occupied little space in the mathematics of selection. A small animal that survived long enough to produce offspring had already fulfilled the requirement that mattered most to inheritance. What happened afterward carried less influence across generations. Repair systems only needed to preserve the body through the dangerous interval before reproduction. Anything beyond that was exposed to weaker evolutionary pressure. Human history followed a similar pattern until very recently. Among hunter-gatherer populations, average lifespan is often estimated around 30 to 40 years, shaped heavily by injury, infection, childbirth, starvation, and violence. Some individuals lived longer, sometimes much longer, yet the average was pulled downward by hazards that modern medicine now prevents or treats. In ancient Rome, life expectancy at birth was roughly 25 to 35 years, deeply affected by infant and childhood mortality. Reaching 70 or 80 years was possible, but uncommon enough to be remembered. The modern world altered that timeline with astonishing speed. Sanitation reduced infectious disease. Vaccines changed childhood survival. Antibiotics transformed once fatal infections into treatable conditions. Surgery, emergency medicine, safer childbirth, and public health systems removed many of the old causes of early death. Within only a few generations, large numbers of humans began living into their 70s, 80s, and 90s as ordinary members of society. Evolution has had almost no time to respond to that shift. Biological change through natural selection usually requires many generations under consistent pressure. Modern longevity expanded too quickly for the genome to be redesigned around it. We are living on a new schedule with older maintenance machinery. This mismatch appears throughout aging. Deoxyribonucleic acid repair pathways that manage damage effectively in youth begin losing precision later. Protein quality control systems that once kept cells clear of molecular debris become less reliable. Stem cell populations that renew tissues diminish with repeated use. Immune responses lose balance, weakening against infection while producing more chronic inflammation. Blood vessels stiffen, muscle declines, bones thin, nerve cells become increasingly vulnerable. These failures do not mean the body is poorly made. They reveal a body being asked to perform beyond the interval that shaped its design. A maintenance program sufficient for 30 or 40 years faces the demands of 80 or 90. Systems that once only needed to carry an animal through reproduction must now sustain identity, memory, movement, and repair across many additional decades. The diseases associated with aging, cancer, cardiovascular disease, neurodegeneration, immune decline, and musculoskeletal deterioration arise partly from this extended use. They are the consequences of running inherited biology far past its original calibration. Modern humans are living inside a rare evolutionary experiment supported by medicine, culture, and technology, while their cells still carry the assumptions of a world where survival beyond reproduction was never guaranteed. A scar is one of the clearest signatures of mammalian repair. It closes a wound, seals danger away, and preserves life, yet it also marks the limit of what the body can rebuild. The tissue survives, but the original architecture is rarely restored. In that difference between closure and true reconstruction, the regeneration gap becomes visible. Across other vertebrate lineages, repair can follow a more complete path. Axolotls, salamanders native to Mexico, can regrow entire limbs after injury. Bone, muscle, nerves, blood vessels, and skin return in organized form. The new structure is functional, connected, and patterned with astonishing precision. The animal does not merely cover the damage, it rebuilds what was lost. Other examples widen the comparison. Zebrafish can regenerate substantial portions of heart muscle after injury, restoring tissue that mammals usually replace with scar. Some salamanders can regrow parts of the spinal cord, sections of the jaw, and even tissues of the eye. Lizards can detach their tails and grow replacements, with cartilage forming where original bone once existed. The result is imperfect compared with the first tail, yet it remains a real working structure. Mammals live under narrower rules. A lost human limb does not return. A damaged spinal cord rarely rebuilds functional connections across the injury. After a heart attack, dead cardiac muscle is largely replaced by scar tissue, and the heart must continue beating with reduced strength. The liver is a famous exception because it can regrow substantial lost mass, yet that ability is specialized. It does not represent a general mammalian capacity to reconstruct complex structures. The difference lies in cellular behavior. Regeneration requires cells to reopen developmental programs, communicate positional information, control inflammation, prevent chaotic growth, and rebuild multiple tissues in correct order. Such systems demand coordination and sustained investment. They also carry risks. A body that encourages extensive cell growth must control that growth carefully, or repair begins to resemble tumor formation. For animals that lived longer lives or faced injuries from which recovery could still matter, maintaining such systems could remain useful. For early mammals shaped by short life histories, the calculation was harsher. Energy spent preserving deep regenerative programs offered little benefit if most individuals died before major repair could unfold. Survival favored fast reproduction and immediate function over the costly preservation of long-term rebuilding capacity. This does not mean mammals lost every trace of regenerative biology. Skin heals, bone can mend, blood renews constantly. Intestinal lining replaces itself at high speed. These forms of repair are real and essential, but they operate within boundaries. They maintain, patch, and replace cells. They do not usually recreate an entire lost structure with all its original complexity. The body remembers fragments of the old language, while the fuller grammar of reconstruction is mostly absent. Researchers studying axolotls and salamanders are therefore looking at more than unusual animals. They are examining a biological capability that reaches deep into vertebrate history. Somewhere along the mammalian path, the full version of that capacity narrowed. The bottleneck offers a reason for why such an expensive gift could fade. In a world where life was brief, rebuilding the future mattered less than reaching the next generation. Many laboratories study aging by looking forward. They test medicines, measure cells, compare tissues, and search for interventions that might extend healthy life. Yet, some of the mechanisms they examine may also be pointing backward toward evolutionary pressures far older than medicine itself. Telomere research offers one example. Telomeres are protective repeat sequences at the ends of chromosomes. Each time many cells divide, these caps tend to shorten. When they become critically short, the cell may stop dividing, malfunction, or die. This discovery became a landmark in aging biology because it linked cellular division to a measurable biological clock. Within mammals, telomere behavior fits the deeper pattern of fast life histories. A body with high cellular turnover spends its telomere reserves more quickly. Small, high metabolism mammals often show shorter lifespans and more rapid cellular cycling. Long-lived reptiles of comparable size can follow a different pace. The contrast reflects more than physiology alone. It also echoes the old evolutionary bargain that favored rapid growth, early reproduction, and compressed maintenance. Senolytic research follows another path into the same story. Senescent cells are damaged cells that stop dividing but remain active inside tissues. At first, this can be protective, especially when a cell might otherwise become cancerous. Across a longer life, however, senescent cells release inflammatory signals that harm nearby tissue. They become biological residue from old repair decisions. Senolytic drugs are designed to remove some of these cells. In mouse studies, clearing senescent cells periodically has improved health span and reduced several age-associated problems. Human trials are now exploring whether similar approaches can help with conditions such as kidney disease, lung scarring, and metabolic complications. Beneath the technical details lies a familiar theme. The treatment attempts to reduce damage left behind by maintenance systems that were never built for long use. Rapamycin brings the pattern into sharper focus. This compound influences a pathway known as mechanistic target of rapamycin, one of the body's major regulators of growth, nutrient sensing, and repair. When this pathway strongly favors growth, cells prioritize building and reproduction-related activity. When its activity is moderated, more resources can shift toward maintenance, stress resistance, and cellular cleanup. In mice, rapamycin has extended lifespan even when treatment begins in middle age. That result matters because it suggests aging can be altered after development is complete. Through the bottleneck lens, rapamycin appears as a way of adjusting an old trade-off. For 164 million years, selection pushed mammals toward growth and reproduction under dangerous conditions. Rapamycin partly tilts that balance toward upkeep. Caloric restriction works through related biological terrain. Across many mammalian studies, reduced calorie intake without malnutrition has been among the most consistent interventions for extending lifespan. It changes nutrient sensing, lowers growth signals, improves cellular maintenance, and reduces certain forms of damage accumulation. Much of this research began without reference to dinosaurs. Scientists were studying chromosomes, damaged cells, nutrient signals, inflammation, and repair. The longevity bottleneck does not replace those discoveries. It gives them a deeper setting. Modern aging science may be trying, piece by piece, to compensate for decisions evolution made in a world where lasting maintenance was rarely rewarded. Aging rarely arrives as a single failure. It moves through the body as a broad shift, touching tissues that perform entirely different jobs, yet seem to follow a similar timetable. The heart, the immune system, the skeleton, the brain, and the cells that replenish worn tissues all begin changing within the same general span of life. Medicine often studies these changes separately. Evolution suggests they may share a common origin. The cardiovascular system gradually loses flexibility. Arteries stiffen, making it harder to absorb the pulse generated by each heartbeat. Bones surrender density as the balance between construction and breakdown drifts. Skeletal muscle shrinks despite continued use. Hormonal signals that once coordinated growth, reproduction, and repair become less stable. Within the nervous system, neurons accumulate damage that is repaired less efficiently than before. At the same time, stem cell populations decline in both number and performance, reducing the body's ability to replace aging tissue. Viewed individually, each process appears to have its own explanation. Together, they form a pattern. Many of the systems responsible for maintenance begin weakening within the same broad period because they were shaped under the same evolutionary conditions. Their schedules were written together. The longevity bottleneck offers a framework for understanding this synchrony. For more than 164 million years, mammalian ancestors lived under pressures that rewarded successful reproduction over prolonged survival. Natural selection refined bodies that could grow, mature, reproduce, and protect themselves long enough to pass genes forward. Maintenance beyond that window brought little advantage because relatively few individuals survived long enough for improvements in extreme old age to influence evolutionary outcomes. As a result, repair programs throughout the body became calibrated to a similar horizon. Deoxyribonucleic acid repair systems, stem cell reserves, immune regulation, tissue renewal, and metabolic maintenance were not optimized independently for maximum durability. They evolved as parts of one integrated organism, serving a life history centered on reproduction and near-term survival. Evolutionary biologists describe part of this process through antagonistic pleiotropy. A single genetic program can provide benefits early in life while generating costs later. Genes that encourage rapid growth, efficient fertility, and strong energy allocation during youth can also increase inflammation, cellular senescence, tissue degeneration, or vulnerability to disease in advanced age. Selection favored the immediate advantages because those benefits improved reproductive success. The delayed consequences existed beyond the primary reach of evolutionary pressure. They accumulated in the background, largely unexamined by natural selection, because few individuals experienced them. That history helps explain why aging appears coordinated across so many organs. The body is not a collection of separate clocks that happen to strike together by coincidence. Many of those clocks were assembled under the same conditions and wound to similar durations. When maintenance systems begin losing effectiveness, the effects emerge across the organism rather than within a single isolated structure. Modern humans now live far beyond the span that shaped much of mammalian biology. Sanitation, nutrition, medicine, and technology have extended life into decades that our ancestors rarely reached. The synchronized decline visible in old age reflects the architecture of a body whose maintenance programs were tuned long ago under pressures shared across an entire lineage. Their timing remains linked because their history remains linked. A shelf of preserved histories still surrounds us. They move through wetlands, bask on riverbanks, patrol coastlines, and drift through island forests. For generations, reptiles and amphibians were often treated as evolutionary side branches, interesting for their uniqueness, yet distant from the problems of human biology. A different picture has begun to emerge. Some of these animals may retain biological traits that mammals gradually surrendered during the long interval when survival depended on speed, reproduction, and brief lifespans. Researchers studying aging increasingly find themselves looking beyond mammals. The reason is straightforward. Certain reptiles and amphibians display patterns of aging that differ dramatically from those seen in humans, dogs, mice, and nearly every other mammal. Mortality rates in some turtle species remain surprisingly stable across long portions of life. Physiological decline can proceed at a pace that appears unusually restrained when compared with mammals of comparable size. Such observations have transformed these animals from biological curiosities into valuable sources of comparison. Among the most revealing findings are studies examining cellular maintenance. Several turtle species maintain strong activity in systems associated with detecting and responding to deoxyribonucleic acid damage, even at ages that would correspond to advanced life stages in mammals. Mammalian tissues often show a gradual reduction in maintenance efficiency as adulthood progresses. Turtles demonstrate a different pattern, one suggesting that repair capacity can remain active for far longer than mammalian biology typically permits. Their cells accumulate damage more gradually, allowing tissues to preserve function across extended spans of time. The tuatara offers another striking example. Found only in New Zealand, this reptile represents the last surviving member of a lineage that flourished long before mammals became dominant. Genetic studies reveal an unusually elaborate collection of genes involved in cellular stress management, temperature response, and protection against oxidative damage. Researchers have identified 37 copies of transient receptor, potential ion channel genes, and 26 selenoprotein genes associated with maintenance and defense against cellular deterioration. Such complexity suggests that some vertebrate lineages retained extensive biological investment in long-term preservation. These discoveries have influenced the direction of modern longevity research, comparative genomics, the practice of comparing complete genetic systems across species, seeks to identify mechanisms present in exceptionally long-lived organisms and determine whether similar pathways could be strengthened or reactivated in mammals. Investigations into bowhead whales, turtles, reptiles, and other durable species increasingly follow this strategy. Scientists compare maintenance programs, repair pathways, and protective mechanisms in search of clues about why aging unfolds so differently across the tree of life. Viewed through the lens of the longevity bottleneck, these animals become more than examples of alternative biology. They function as living reference points. Their genomes preserve information about strategies that remained useful outside the constraints that shaped mammalian evolution. Each comparison helps distinguish which features are universal to vertebrates and which may have been diminished or lost within the mammalian lineage. The field therefore resembles a form of biological archaeology. Instead of excavating fossils from stone, researchers examine living organisms that still carry fragments of older solutions to survival and maintenance. Reptiles and amphibians are no longer standing at the edge of the story. They have become guides, offering glimpses of cellular capabilities that endured while mammalian evolution followed a different path through deep time. Some histories refuse to separate into clean categories. They cannot be placed entirely among losses or entirely among gifts. The mammalian story belongs to that kind of history, because the same pressure that narrowed the body also enlarged the mind that now studies the narrowing. For 164 million years, mammalian ancestors lived under constraints that favored small size, rapid reproduction, night activity, and constant vigilance. Those pressures carried costs already traced through the body. Color vision narrowed, photolyase disappeared, long-term repair systems weakened, regeneration diminished. Aging became faster than it might have been under another evolutionary path. Yet those same conditions demanded more from the nervous system. An animal active in darkness could not rely on vision alone. It needed scent, sound, touch, memory, and spatial awareness to combine into action. It needed to recognize patterns in incomplete information. It needed to distinguish danger from opportunity without the full clarity of daylight. Every successful generation passed forward nervous systems capable of managing that sensory world with increasing precision. From that pressure came the expanding mammalian brain. Olfactory regions grew more complex, hearing became more refined. The cerebellum and forebrain changed in ways suited to movement, learning, and rapid adjustment. The neocortex, that layered outer region responsible for integrating experience, became one of the defining structures of mammalian life. In its earliest forms, this expansion helped small animals survive. Much later, across many branches and many experiments in mammalian evolution, it produced social memory, flexible behavior, parental care, tool use, symbolic thought, and eventually human language. The path was indirect, contingent, and filled with extinct side routes. Still, the connection remains. The pressures that made mammalian life shorter also helped build the neural architecture that made mammalian thought deeper. This is the paradox at the center of the story. The bottleneck stripped away parts of the body's long-term maintenance, while also shaping the organ capable of understanding long-term history. The same evolutionary darkness that reduced repair capacity helped create a brain able to study repair. The same lineage that lost photolyase eventually produced laboratories where that loss could be detected, measured, and partly reversed in mice. There is no clean moral inside that arrangement. Evolution does not trade in fairness. It produces outcomes from pressure, inheritance, accident, and time. A trait that begins as a survival response may become the foundation for something far beyond its first function. A limitation may persist beside an astonishing capacity. A cost may travel with a gift until they become impossible to untangle. The mind following this history is not outside the bottleneck, it is one of its products. Its curiosity, memory, and ability to imagine extinct worlds all grew from the same inheritance that shaped its aging cells. The body carries the cost of the compression, and the brain carries the means to recognize it. That is why the story does not close neatly around loss. Mammals were diminished by the age of dinosaurs, yet they were also transformed by it. Out of that transformation came a creature capable of reading stone, comparing genomes, naming vanished enzymes, and discovering that its own mortality has a history older than humanity itself. A band of clay, no thicker than a finger, marks one of the sharpest turning points in Earth's history. It lies buried within sediments on multiple continents, carrying an unusual abundance of iridium, an element far more common in asteroids than in the crust beneath our feet. Wherever geologists find that layer, they find the same dividing line. One world ends beneath it, another begins above it. The rocks tell the story with startling clarity. Fossils of non-avian dinosaurs fill the layers below, move upward across that narrow boundary, and their presence vanishes. Entire ecosystems disappear from the record. Oceans change, forests change, food webs rearrange themselves. A planetary chapter closes within a geological instant. Yet stone preserves only part of the inheritance. Running beside that visible boundary is another, one that cannot be seen in cliffs or quarries. It exists inside living cells. It is carried through chromosomes, proteins, and developmental pathways. Every mammal alive today stands above the iridium layer in time, while carrying biological traces of pressures that accumulated long before the asteroid arrived. Those traces are not dramatic scars. They appear as absences, constraints, and compromises. A repair enzyme, once common among vertebrates, disappeared during the Long Age when mammalian ancestors lived under relentless ecological compression. The broader palette of color perception carried by distant forebears narrowed and never fully returned. Regenerative abilities faded. Maintenance systems that support exceptional longevity weakened generation after generation. The asteroid ended the reign of the dinosaurs, but it did not erase the biological consequences already written into mammalian evolution. Look closely at any modern mammal, and those consequences remain present. The skin confronting sunlight relies on repair mechanisms shaped by losses that occurred deep in the Mesozoic era. The aging process unfolds through maintenance systems calibrated under conditions very different from those experienced by modern humans. The body carries solutions crafted for survival in a demanding world populated by dominant reptilian competitors. Evolution rarely restores every discarded capability once the original pressure disappears. This continuity stretches across distances almost impossible to grasp. Before the first primate climbed through a forest canopy, before grasses spread across vast landscapes, before continents settled into their modern arrangement, the foundations of mammalian biology were already being molded. Subsequent ages added refinements, innovations, and adaptations, yet many deeper constraints remained embedded beneath those newer layers. There is nothing deliberate about this inheritance. Dinosaurs did not shape mammals through intention. Predators pursued food, prey sought shelter. Natural selection operated without foresight, preserving traits that improved immediate survival and allowing others to fade when their costs exceeded their benefits. Across millions of generations, those small decisions accumulated into a biological architecture that still surrounds every mammalian cell. The clay boundary records a moment of catastrophe. The genome records a far longer narrative, written gradually through countless breeding seasons and countless encounters between hunter and hunted. Together, they form two versions of the same history. One is etched into stone and visible to the eye. The other remains embedded within living bodies, carried forward across time, a debt inherited from a vanished world and paid by descendants that never saw it. Across laboratories, hospitals, and research institutes, a remarkable effort is unfolding. For most of evolutionary history, natural selection shaped life through countless generations, altering bodies only through inheritance and time. Now, for the first time, a species is attempting to intervene directly in the aging process itself, examining limitations carried for more than 100 million years and asking whether some of them can be modified, bypassed, or repaired. One avenue of investigation focuses on senescent cells. These are cells that have stopped dividing after accumulating damage, yet remain active within tissues. As years pass, they release signals that contribute to inflammation and disrupt the function of neighboring cells. Scientists have developed experimental compounds known as senolytics, designed to identify and eliminate these lingering cells. Studies in mice have produced encouraging results, extending healthy lifespan and reducing multiple age related disorders. Human clinical trials are now exploring their potential use in conditions such as kidney disease, pulmonary fibrosis, and complications associated with diabetes. Another major field centers on a molecular pathway known as mechanistic target of rapamycin. This pathway helps determine how cells allocate resources between growth and maintenance. During youth, strong growth signals support development and reproduction. Later in life, those same signals can contribute to deterioration. A compound called rapamycin has repeatedly extended lifespan in laboratory mice, even when treatment begins during middle age. Researchers are investigating dosing strategies that might preserve benefits while minimizing unwanted effects on immune function. The goal is not immortality, it is the possibility of maintaining tissue integrity for longer periods. Attention has also turned toward nicotinamide adenine dinucleotide, a molecule involved in energy, metabolism, and cellular repair processes. Levels decline with age across many tissues. Various precursor compounds are being studied as a means of restoring those levels, potentially improving cellular maintenance and supporting repair pathways that become less effective over time. Evidence remains under active evaluation, yet the effort reflects a broader theme running through modern geroscience, reinforcing systems that gradually lose efficiency with age. The story of photolyase has entered this landscape as well. Researchers are developing topical formulations that deliver the enzyme directly to skin cells. In this context, the objective is straightforward: repairing ultraviolet induced damage using a tool absent from mammalian biology. Some scientists have also begun exploring whether future technologies might safely restore similar capabilities within specific tissues. Such work remains highly experimental, but it illustrates how deeply evolutionary history now informs biomedical thinking. Comparative genomics has opened another path. Investigators examine the genomes of bowhead whales, tuataras, naked mole rats, and long-lived turtles, searching for features associated with exceptional maintenance, cancer resistance, and resilience. By comparing these species with humans and other mammals, researchers hope to identify biological mechanisms that could someday be activated, strengthened, or imitated. None of these approaches carries a guarantee. Many promising ideas fail during testing. Biological systems are interconnected, and interventions that succeed in mice do not always succeed in humans. Even so, the direction is unmistakable. A mammal now possesses the knowledge to recognize inherited constraints, trace their origins through deep time, and attempt, with caution and patience, to reshape some of the rules written long ago into its cells. A strand of gray hair seems like such a small thing. It appears one morning among darker strands, almost unnoticed at first. Years later, there are more of them. Eventually, they become part of the face in the mirror, accepted as one of the ordinary signatures of age. Yet within that change lies a story that reaches far beyond any individual life, extending backward through deep evolutionary time. The color of hair depends on specialized cells called melanocytes and on stem cells that replenish them. Across the decades, those stem cells gradually diminish, their reserves shrink. Fewer pigment-producing cells replace those that are lost. The hair continues to grow, but color fades from the process. Modern biology describes the mechanism in detail. The broader evolutionary context suggests why such depletion occurs at all. Mammalian maintenance systems were shaped around a limited lifespan, one in which preserving stem cell populations far into old age offered little advantage under the conditions that dominated much of mammalian history. The same pattern emerges in the joints. Cartilage cushions movement, absorbs force, and allows bones to glide past one another. Unlike many tissues, it possesses only a limited capacity for repair. Small injuries accumulate, microscopic wear remains, decade follows decade, and the burden grows. By later life, stiffness and discomfort often accompany motions that once seemed effortless. This limitation reflects a lineage whose regenerative capabilities became progressively reduced, leaving mammals with fewer options for rebuilding complex structures after damage occurs. Skin tells a similar story. Each day, sunlight leaves traces within cellular deoxyribonucleic acid. Repair systems correct much of that damage, preserving function across countless exposures. Yet one important repair tool vanished from the mammalian lineage long ago. Without it, cells rely on alternative pathways that perform their task effectively but imperfectly. The marks that remain accumulate across years. Wrinkles deepen. Elasticity declines. The passage of time becomes visible on the body's surface through processes linked to molecular events rooted in a distant evolutionary past. Cancer follows the same logic, though with far greater consequences. Throughout life, mutations arise in dividing cells. Most are corrected, contained, or rendered harmless. Some escape detection. Additional changes accumulate. Protective systems intervene repeatedly, often with extraordinary success. Eventually, in certain cells, enough alterations gather to bypass those safeguards. Uncontrolled growth begins. The disease appears late in life because it often requires many years of accumulated errors. Its origins lie within the same balance between repair, maintenance, and survival that shaped mammalian biology under long-standing evolutionary pressures. These connections are not symbolic. They emerge from documented mechanisms, measurable pathways, and observable genetic history. The chain linking present biology to ancient selection pressures can be followed step by step through comparative genomics, cellular biology, and evolutionary theory. Seen from this perspective, an aging body becomes a record of events far older than memory. Every fading pigment cell, every persistent scar, every exhausted repair pathway carries information from a vanished world. Long after the asteroid ended the age of dinosaurs, the biological consequences of living alongside them remained. They persist within tissues, organs, and cells, preserving a history written not in books or stone, but in the workings of life itself. The deepest consequences of history are often the ones that leave no visible monument behind. Mountains reveal collisions, fossils reveal extinction. Layers of rock preserve floods, droughts, impacts, and vanished oceans. Yet some events leave their records somewhere else entirely, written into living tissue, carried forward through inheritance long after the world that created them has disappeared. The dinosaurs are gone. They have been gone for 66 million years. Their skeletons have become stone. Their nesting grounds are buried beneath layers of sediment. The forests they crossed have been transformed into coal. The shallow seas, where many hunted and fed, have hardened into limestone. Entire ecosystems have passed from life into geology. Viewed from the perspective of ordinary time, their disappearance seems complete. No living mammal has encountered a non-avian dinosaur. No culture remembers them from direct experience. Their age belongs to deep history, separated from the present by a span of time so immense that human civilization occupies only the final fraction of it. And yet, the biological effects of their dominance remain active. They survive in the architecture of mammalian cells. They survive in repair systems operating under limitations established during the long Mesozoic compression. They survive in aging patterns, regenerative constraints, cancer vulnerability, and the gradual accumulation of molecular damage. The animals themselves vanished, but the evolutionary pressures they imposed continued forward inside the descendants of the creatures that endured alongside them. This is evolutionary debt in its purest form. A lineage adapts to survive under a specific set of conditions. Genes are lost, pathways are reduced, investments shift from maintenance toward reproduction. Cellular priorities are rewritten across millions of generations. When the original pressure disappears, those changes do not automatically reverse. Evolution possesses no memory of previous states and no mechanism for restoring what once existed merely because circumstances have changed. For that reason, 66 million years proved insufficient to fully undo what 164 million years had installed. Mammals diversified into forms their ancestors could never have imagined. Some entered the oceans and became whales. Others mastered powered flight. Some grew into giant herbivores. Others developed elaborate social structures and extraordinary intelligence. Primates emerged, and among them appeared a lineage capable of language, science, and historical reconstruction. Across all of that transformation, the deeper framework remained in place. Every whale, every bat, every elephant, every mouse, every human carries traces of the bottleneck. Their bodies still operate within biological boundaries shaped during an age dominated by enormous predators. Their genomes still bear evidence of ancient compromises. Their cells still reflect decisions made by natural selection in a world that no longer exists. What makes this realization unusual is that the shadow is measurable. It can be traced through comparative genomics. It can be observed in repair pathways, longevity differences, and regenerative capacities. It appears in published data, laboratory experiments, and evolutionary reconstructions. The connection is not symbolic, it is biological. The shadow did not end when the asteroid struck. It continued through every generation that followed, passing from ancestor to descendant without interruption. The dinosaurs became dust, stone, and memory. Their influence remained alive, carried forward in mammalian biology, still shaping bodies that walk the earth today. The age of dinosaurs ended. The consequences of that age never fully did. At the edge of every scientific explanation sits a boundary where certainty begins to thin. The longevity bottleneck hypothesis reaches such a boundary. It offers a coherent account of how mammalian aging may have been shaped by 164 million years of evolutionary compression, linking losses in repair, regeneration, and maintenance to a world dominated by giant predators. Yet one question remains suspended above everything that has come before. How much of what was lost can truly be recovered? The evidence gathered so far provides reasons for both caution and possibility. Among the most striking examples is photolyase. For tens of millions of years, mammals lived without the enzyme that directly repairs ultraviolet damage in deoxyribonucleic acid. The gene disappeared, yet experiments have demonstrated that mammalian cells can still use the enzyme when it is supplied. The surrounding biological machinery remains capable of cooperating with it. A tool once absent can function again. That discovery does not solve aging, but it reveals an important principle. Some evolutionary losses may leave behind foundations that never entirely vanished. Photolyase, however, represents only a single thread in a much larger tapestry. The bottleneck was not the removal of one gene or one pathway. It unfolded across immense spans of time, influencing networks of interacting systems. Repair mechanisms, cellular maintenance programs, regenerative responses, metabolic priorities, and stress resistance pathways all experienced selection pressures that favored immediate survival and reproduction. Untangling those changes requires reading a biological record written across entire genomes. Researchers are still attempting to map that record in full. Comparative genomics continues to reveal differences between mammals and species that age more slowly or maintain greater regenerative capacity. Each newly identified pathway raises another possibility. Some missing functions may prove relatively straightforward to restore if the underlying cellular framework remains intact. Other alterations appear woven into the deepest architecture of mammalian biology, affecting how energy is distributed between growth, reproduction, defense, and maintenance throughout life. That distinction matters because replacing a missing component differs greatly from redesigning an integrated system. A cell can sometimes accept a restored tool. Rebalancing millions of years of evolutionary priorities presents a more complex challenge. Biological systems are interconnected. A change that improves one process may influence many others. Repair, growth, immunity, fertility, and metabolism participate in a network shaped by deep history. Adjusting one strand can affect the entire pattern. Beyond the laboratory stands another layer of uncertainty. Extending lifespan is not solely a technical problem. It also touches questions about how a species relates to its own evolutionary inheritance. Human beings already live far beyond the conditions under which mammalian biology was originally calibrated. If future discoveries push that boundary further, the consequences will reach beyond cells and tissues into culture, memory, and the structure of human life itself. For now, the answers remain incomplete. Laboratories across the world continue to investigate aging, longevity, repair, and regeneration. New tools appear each year. New genomes are sequenced. New pathways are uncovered. The work advances through the same enlarged brain that emerged from the history described throughout this story. A lineage shaped by constraint has become capable of examining the constraint itself, searching through its own biology to discover what remains, what was lost, and what might still be found again. Far back in the Mesozoic night, a small, warm-blooded animal waited below ground, while heavier bodies moved through the world above. It had no language, no history, no imagined future. Its life was made of scent, hunger, warmth, danger, mating, shelter, and the next breath. Every instinct served a narrow purpose: to remain alive long enough for another generation to begin. That animal was not human in any recognizable way. Its body was low to the ground. Its senses were tuned to darkness. Its lifespan was brief. Its world was immediate. Yet across an unbroken chain of reproduction, it belongs to the same lineage that eventually produced every person who has ever lived. Between that burrow and the modern human body lies an almost unimaginable sequence of births, each one carrying forward whatever had helped the previous generation survive. This is the intimacy of evolution. An ancestor does not need to resemble its descendant to be real. The connection is biological, continuous, and physical. Every human cell carries inheritance from animals that survived the dinosaur age by staying small moving carefully, reproducing early, and yielding much of long-term maintenance to the demands of immediate survival. The body you inhabit is not separate from that history. It is one of its latest expressions. The costs remain within that inheritance. The missing photolyase, the narrowed visual legacy, the weakened capacity for full regeneration, the repair systems that work, yet leave gaps, the aging clock that moves faster than the clocks of turtles, tuataras, and other lineages that never passed through the same compression. These are not distant curiosities. They are active features of mammalian biology, present in skin, blood, nerves, organs, and the slow wear of time. Yet the same inheritance contains something else. The pressure that favored darkness also favored sensory integration. Smell, hearing, touch, memory, and movement had to work together under uncertain conditions. Across millions of generations, those demands helped shape the mammalian brain. The neocortex expanded. Behavior became more flexible. Learning became more powerful. In one primate branch, that long history eventually led to symbolic thought, language, music, mathematics, grief, tenderness, and science. The small animal in the burrow did not aim toward any of this. Evolution has no distant intention. It did not survive in order to become human. It survived because survival itself was enough. Yet when survival continues across enough generations, under enough pressure, new possibilities accumulate. What began as a life built around evasion and reproduction eventually produced a mind capable of reconstructing the very conditions that shaped it. That is the strange tenderness of this story. The same cells that carry compromised repair systems also support memory and imagination. The same lineage marked by accelerated aging produced beings able to understand aging as history. The body holds the debt while the mind reads the record. Something small endured the age of dinosaurs. It passed through darkness, hunger, fear, and compression. Its descendants filled the world. Then one of them learned to look backward and recognize the cost of survival written into its own biology. There is a tendency, when looking backward across immense spans of time, to search for a verdict, to decide whether the story has been tragic or fortunate, whether the losses outweigh the gains, whether the path that led here should be understood as a burden or a triumph. The history uncovered by the Longevity Bottleneck Hypothesis resists such simplicity. It presents a record of costs that are real, benefits that are real, and a future that remains unwritten. The curse is real. It exists in laboratory measurements, genomic comparisons, and cellular biology. Photolyase disappeared from the mammalian lineage. Longevity systems that remain active in some reptiles and other vertebrates became reduced or altered. Regenerative capacities narrowed. Maintenance programs that could have preserved tissues for longer periods yielded ground to strategies centered on rapid reproduction and survival under constant threat. The aging clock inherited by mammals was calibrated within a world that demanded immediate success, not exceptional longevity. Those consequences still accompany every mammalian body. Cells accumulate damage. Repair systems operate with limits. Stem cell populations decline with age. Regeneration remains partial. Cancer emerges from mutations that evade correction. These are not philosophical ideas. They are biological realities observed repeatedly across species, tissues, and generations. The marks of the bottleneck remain embedded within the architecture of mammalian life. Yet the gift is equally undeniable. The same evolutionary pressures that narrowed some possibilities expanded others. Neural processing grew more sophisticated. Sensory information became increasingly integrated. Learning gained greater importance. Memory acquired new depth. Across millions of generations, those developments contributed to the emergence of the mammalian brain and, eventually, the human mind. That mind now performs an extraordinary act. It studies the very history that shaped it. It extracts deoxyribonucleic acid from fossils. It reconstructs ancestral genomes. It compares turtles, whales, salamanders, tuataras, naked mole rats, and humans. It identifies missing enzymes and degraded pathways. It uncovers events buried across geological ages and translates them into knowledge. The pressure that constrained the body also helped create the intelligence capable of recognizing the constraint. For the first time in Earth's history, a species is attempting to understand the evolutionary origins of its own limitations and determine whether any of them can be changed. Photolyase restoration experiments demonstrated that at least some lost functions remain biologically compatible with mammalian cells. Longevity research continues to explore cellular maintenance, regenerative medicine, comparative genomics, nutrient sensing pathways, and mechanisms of damage repair. Each discovery adds another small piece to a puzzle assembled across deep time. No outcome is guaranteed. Some approaches may fail completely, others may produce modest benefits. Certain constraints may prove more deeply rooted than researchers currently imagine. Biology is intricate, and every intervention carries consequences that must be understood with care. Even so, the direction of inquiry itself represents something new. The bottleneck was created by evolutionary pressures acting over 164 million years. It shaped bodies that endured, adapted, and survived. From those bodies emerged minds capable of reflection, investigation, and deliberate action. The dinosaurs are gone. The shadow they cast remains written into mammalian biology. Yet within that shadow stands a descendant of the small animal that once waited in darkness, carrying both the inheritance of the wound and the growing possibility of repair.